Okay, good morning, everybody. And today I'm going to just finish off our discussion about circulation and gas exchange, and then segue into something intimately involved with our discussion about circulation, which is the regulation, osmoregulation in animals, the control of water and solutes. So before I begin, please remember that next Wednesday you have your second test. And I very, very strongly recommend that you go and do the assignments, especially the latest assignment which I have put up. I go to the assignments to look up the scores because I need to do the scores, needed the assignment scores for your assessment, your midterm assessment, and I find that less than half of the class have completed assignments. This is extremely foolish because you are throwing away grade points. You're throwing them away by not doing the assignments. And you are missing an opportunity for revision, for revising the material. And it is quite apparent from the grades that I have obtained so far that most of the class are not bothering to keep up with their studies, with their revision. You're still leaving everything till the night before tests and quizzes to try and cover an enormous amount of material, of complex material. This material requires work. It requires hard work. And if you don't do the hard work, you're not going to get the grades. And it's as simple as that. So please, you now have a week to go before your next test. Can you please make an effort to study properly for this next test, because it is critical to most of you that you get a good grade. Okay, and that is all that I'm going to say about that at the moment. Let's return to our the final slides for our section on circulation, because we have discussed at length how a major responsibility of the circulatory system is to conduct gases around the organism. And that is to take oxygen to even the remotest tissues and gather up waste carbon dioxide and conduct it back to the lungs to be expelled. Now, in order to do that, we first will need an efficient liquid-based circulatory system. But we also need to realize that if these gases, both oxygen and carbon dioxide, were simply to dissolve into the water component of the blood and then be circulated, it would be an extremely inefficient system. It would be too inefficient that for survival of the organism. Instead, some kind of accessory system has to be adopted. And this takes the form of two things. The first is that there are a set of respiratory pigments which can bind, in particular bind oxygen, can also bind carbon dioxide to a small extent. And second of all, there is a special system for transporting carbon dioxide. So there's a specialized system for transporting oxygen and a small amount of carbon dioxide. Those are the respiratory pigments. And then carbon dioxide is transported in the form of an ion, which is bicarbonate. So it's surely familiar to you. Sorry, let me just do this. Oh, I want to do this. There we go. It is surely familiar to you that the major respiratory pigment in our blood is hemoglobin. But that hemoglobin is not the only respiratory pigment in the animal kingdom. There are others. For example, arthropods and mollusks have a compound called hemocyanin. It has nothing to do with cyanide. It's just because it's blue. And hemocyanin has copper as the oxygen binding ion, metal ion. In our hemoglobin, of course, the oxygen binding metal ion is iron. And that is the structure of hemoglobin is actually 4 proteins, 4 hemoglobin molecules, which are joined together to form a single hemoglobin compound. Here it is here. This is its, in terms of protein structure, this is its quaternary structure. That is, it consists of 4 different modules, if you like, which, or subunits, 2 here, 1 there, 1 there, 1 there, and 1 there. And you can see where they are, because here in the center here is the heme group. And the heme group is what contains the ion. And so it's here in the heme group that oxygens are bound. Each whole hemoglobin molecule can bind 4 atoms of oxygen. And that this occurs in a kind of a strange way. If you look at the way in which it's bound, initially, if one oxygen binds here, it makes it much easier for the other hemoglobin units to bind oxygen. 2 bind, it enables these 2. 3 bind, it strengthens the binding of this. So this is termed cooperative binding. And in a sense, the 4 subunits interact with one another. Now that forms oxyhemoglobin. And oxyhemoglobin is the form in which oxygen is transported around the body. The oxyhemoglobin sticks tightly to the hemoglobin until it reaches an area where there is a very low concentration of oxygen. And the hemoglobin loses its ability to bind the oxygen, and the oxygen is dumped, and it diffuses to the tissues. The hemoglobin can also bind carbon dioxide and form a compound called carboxyhemoglobin. But this is not an efficient way of transporting carbon dioxide around the body. A very small amount of carbon dioxide can be transported by hemoglobin back to the lungs, where it's dumped and breathed out. But by far the majority of carbon dioxide is actually formed into an ion. The carbon dioxide dissolves in water and it forms carbonic acid, H2CO3, which can dissociate into a proton, H+, and a bicarbonate ion. And it is in this form, the bicarbonate ion, that the majority of our carbon dioxide is transported by the blood back to the lungs, where the conditions in the lungs favor then the formation, the dissolution of the ions back to water and to carbon dioxide. Carbon dioxide is dumped through the walls of the alveolus and it's breathed out. Okay, so that's all that I need to tell you about. There's only one thing I wanted to remind you about. One thing I did forget, oh, I haven't got the other slides, but I'll just tell you. There was one thing I needed to remind you about. We talked extensively about the circulation system, and we saw how the blood goes via the arteries to the body and then through the veins back to the heart. And I needed to remind you that there are in the body some vessels which link organs directly that do not go through the heart for their circulation. And these are called the portal vessels. And the one, we mentioned it before in our discussion about nutrition, because the nutrient absorbed by the intestine, for example, is gathered into a portal vessel, the hepatic portal vessel, which transports blood directly from the intestine to the liver. It doesn't go through the heart. Any vessel which does that, which links two organs without going through the heart, is termed a portal vessel. Okay, so now let's move on to a discussion about osmoregulation and excretion. A very brief discussion, I might add. The reason that I said before that this was intimately concerned with circulation is because what we are dealing with in both cases in circulation, we're talking about transport via a fluid around the body and around the cells. We saw how the body fluids, the serum, leaves the capillaries and bathes the tissues and is then gathered back up as lymph and returned to the circulatory system. In both the cells and in our body fluids, we have solutions. We have solutions of different ions, of different compounds, of proteins, of all sorts of things. So it is very possible that we may end up with situations where a cell may be bathed by a solution whose composition differs to the cytoplasm. And this can have important consequences. In addition, an entire organism has a water balance, a solvent balance that it needs to satisfy. It has to keep the solutions in the body at a certain concentration. At the same time, the organism has to void waste. It has to somehow or other get rid of waste. And that is particularly true of the waste which is produced by the metabolism of nitrogenous compounds, proteins, nucleic acids. These sort of things produce toxic waste products, which must be voided. And so an organism has to do balance these two things. They have to be able to have enough water that they are able to void their waste. At the same time, they have to have enough water internally that they maintain a certain osmotic balance. And they may have to maintain different osmotic balances in different organs or in different portions of an organ. So it becomes a very complex issue. At its simplest, let's remind ourselves about some of the situations that we have heard about before. And for all of these, let's picture the cell as being cytoplasm surrounded by a cell membrane. And that cell membrane, the critical feature of it is that it is semi-permeable. That means it allows certain substances to go across and others not. In general, the cell membrane does not allow ions, salts, proteins, these sorts of things to move across very easily. But water can pass quite freely backwards and forwards through special water channels, especially called the aquaporins. So the membrane allows the passage of water, but it usually doesn't allow the passage of salts and solutes, which are dissolved in the water. So this sets up a situation where, for example, if I have a situation on the outside of a cell where there are very few solutes, and there on the inside of the cell, I have a lot. The cell now is said to be hyperosmotic to its surroundings. The surroundings are hypoosmotic to the cell, but the cell is hyperosmotic to its surroundings. Now, the easiest way to think about this is not in terms of the solutes, but in terms of the number of molecules of water that you have, the concentration of water that you have on either side. The concentration of water on this side of the cell is much lower than the concentration of water on that side. There's more stuff, if you like to think of it like that, on the inside here, on this side of the membrane. There's not the room for the water. So the water concentration here is lower than it is here. The tendency of all substances and in normal conditions, if they are free to move, is to move down their concentration gradient. They move from an area of high concentration to an area of low concentration. Exactly the same thing happens here. We've got a high concentration of water here and a low concentration of water there. So water will tend to move in this direction. It will start to dilute this solution because the water is moving from an area of high water concentration to an area of low water concentration. The movement of water by diffusion, it has its own special term, and that is osmosis. That is what osmosis is. It's the movement of water down a diffusion gradient. So water is moving into the cell by osmosis. Now, for most animal cells, this is a very dangerous situation because water floods into the cell begins to expand, the volume begins to increase as the water floods in, and the cell membrane has very little resistance to being stretched like this, and it will eventually burst. So this is a very dangerous situation for all animal cells. Now, a situation where we have an equal water concentration on either side, we call an isosmotic situation. It's isosmotic or isoosmotic. So in this situation, the water concentration is exactly the same on that side as that side. Does water movement stop? No, it doesn't. The net water movement stops, because if I allow a water molecule to move across this membrane to this side, the water concentration on this side is now higher. And a water molecule is going to move that way, and it will balance on either side. In the isosmotic situation, as many water molecules move across this way, move across that way. They are in balance, and the cell, there is neither gain nor net gain nor loss of water. The opposite situation to the hyperosmotic situation that we saw here is when a cell is hypoosmotic to its surroundings. And under this situation, the water concentration is higher inside the cell than it is outside. Water is going to flood out of the cell, and the cell is going to shrink. Both of these situations, that one there and this one here, are very dangerous for a cell. But remember that we're not just dealing with, or we're usually not just dealing with a cell floating around. We're dealing with an animal, and the animal has an entire set of organ systems for which a major responsibility in an animal is to maintain osmotic balance, to regulate the osmotic conditions to which their cells are exposed as far as possible, to maintain a situation where the cell is in a favorable homeostatic situation, where the movement of water and the movement of solutes are controlled to present cells with a controlled environment. So there are many different strategies which different animals and which different organs, in actual fact, adopt in order to osmoregulate. Almost all invertebrates, for example, don't do anything at all. Marine invertebrates, rather. They are termed osmoconformers. That is, they simply maintain their tissues at the same concentration as the seawater around them. They just they don't have to do anything. Their adaptations are internal to the cytoplasm, dealing with a high salt concentration and continuing to operate. They may still actively transport solutes back and forth, but they do so. That is not a response to changes in osmotic environment. Osmoregulators, on the other hand, are organisms, animals in particular, which expend energy to control their water uptake and their water loss, and to really strictly control their solutes and the disposition of their solutes. It is most common that animals have a very strict osmotic requirement, have very strict osmotic requirements. They cannot be varied very widely one way or the other. And those we call the stenohaline organisms, and that is really the majority of organisms. There are a few which we call euryhaline, which are able to tolerate wide fluctuations in external osmolarity. But those are usually in special situations. For example, the organisms which live in estuarine environments. An estuary is the place where a river meets the sea, and estuarine organisms are adapted to rapid changes in salinity as tides go in and go out. Another interesting one are the salmon, for example, and fish like that, which live in both freshwater and in marine systems. They have to be able to adapt to the two, because those two situations are very, very different to one another, as we'll see in a minute. Okay, so let's just think. This example of fish is a very good one, because fish actually evolved in fresh water, and they have maintained a body concentration, actually, which is quite close to water. And so in this, in both marine and freshwater fish, their body fluids don't actually vary that much from one another, surprisingly enough. Their fish tissue, the fish tissue is about 1.5% salt in both instances. Now, the fish are very strict osmoregulators, and they use, we're going to see that there, throughout these systems, there are two ways in which you can control the movement of water and solutes. The first is by passive transport, right? You just allow substances to diffuse down their concentration gradient. If you want an ion to move in a particular direction, you set up the conditions, for example, where that direction of flow will be maintained. We'll see an example of this when we talk about the kidney. But they also use active transport, and the active transport mechanisms are those which require an input of metabolic energy to move substances against their concentration gradient. And organisms expend a tremendous amount of energy on osmoregulation through active transport, because maintenance of the correct osmotic environment is so critically important. The health of the cells depends very much on maintaining a homeostasis of the Earth's osmotic environment. So let's have a look at these fish in two different situations, remembering that they have much the same osmolarity of their body fluids. The solute concentrations in both is about the same. Now I'm going to take an oceanic fish, and I'm going to have a look at it. Its environment is living in seawater, which is about 3.5% salt. And its tissues are 1.5% salt. What is the situation of the fish tissue? Is it hyperosmotic or hypoosmotic to its environment? In other words, which of these situations are we talking about for the fish's cells bathed by seawater with a solute concentration so much higher than the body fluids of this and cytoplasm of the cell itself? Well, in that situation, the cell is hypoosmotic to its environment. 3.5% the seawater, 1.5% the body fluids in the cytoplasm of the fish cells. What is going to happen in a marine fish is that water is going to continually flood out of those cells. And this is indeed what happens. Water is continuously lost by the fish. And as a result, the fish has to drink continuously. That's the first thing they do. They continuously drink water. And they have special active transport systems in their own kidneys and in the gills, especially, which pump sodium chloride out remove the sodium chloride from the body fluids as fast as possible. So in actual fact, if you want to think about it this way, oceanic fish is in danger of dehydrating because they continually lose water. What about the freshwater fish? We now got the situation. We remember we got 1.5% salt in the body fluids and the cytoplasm of the cells, and the water around it is only 0.5% salt in fresh water. Which of these situations are we now looking at? We're looking at this. We're looking at a situation where the cell is hyperosmotic to its environment. Here's the fresh water outside, very low salt, the very low solutes, and here's the inside of the cells and the inside of the fish, very high. Water is going to continuously flood in. In effect, the freshwater fish is in danger of drowning, of having its cells swell up and burst, but in any case being flooded with water that is continually coming in. The freshwater fish has special mechanisms to void that water as quickly as possible. It has very, very active kidneys, for example, and they continuously void a very dilute urine, which gets rid of the water that is flooding in. In the case of the seawater fish, or the marine fish, they excrete a very concentrated urine, because what they've done is they extract, their kidneys extract as much water as they can before they avoid the urine, and they excrete as much salt as they possibly can. So their urine is extremely concentrated. So those are the two contrasting situations. We now need to think, well, these are aquatic organisms. And what about terrestrial organisms like us? What is our environment like around us? And if you want to think about it, well, what is happening to the water in our bodies the whole time? We are evaporating water out. This is a huge problem for terrestrial organisms, that they run the risk of water actually evaporating from their tissues. In a way, they are hypoosmotic, to their environment in a way, because the water concentration is lower outside the body than it is inside. And terrestrial organisms are in danger of dehydrating. In addition, terrestrial organisms face a difficulty that they have to void their waste. They have to somehow get rid of waste products, especially nitrogenous waste products, which are rather toxic. So these are covered up initially, okay? But if you go to your textbook and just examine these different situations, you can actually move these across, okay, and see what the solutions are. But go to those diagrams and contrast the three situations, the marine, the freshwater, and then terrestrial situation, and think about what are the strategies which each of those organisms has to adopt. First of all, in dealing with water, either getting rid of it or else conserving it. Second of all, in dealing with waste products. How do they deal with their waste products? Because they're going to, especially in the case of terrestrial organisms, they are going to have to somehow get rid of waste without using too much water. In the case of these aquatic organisms, they very frequently produce ammonia as their primary waste product. So just as an example, ammonia is very, very toxic, but it's also very soluble. So if you have enough water available, then you can, in fact, flush it out. You can flush out the ammonia. And that is what both, in actual fact, both cases, marine fish and freshwater fish, and many other aquatic organisms that have abundant water actually in their environment, they will flush out, they'll produce ammonia as their waste product because they can flush it out easily. Terrestrial organisms have to think about something different. They have to actually adopt strategies which will conserve water, which will stop water from evaporating. So there are various ways that this can be done. The first is in anatomy. If you think about things like the arthropods, those kinds of terrestrial organisms, they are covered with a chitinous exoskeleton, which limits the amount of water that they lose. The real problem with loss of water comes, first of all, in dealing with the waste products, but second of all, that all organisms need, at some level or another, they need to transfer gases from the environment to their cells in the form of a dissolved, solute. So, for example, our oxygen moves across from our lungs into a fluid environment. It is bound to hemoglobin. When it reaches the tissues, it passes from the hemoglobin in the solute form across to cells. Same way, carbon dioxide leaves cells. It is dissolved into the fluids of the circulatory system. Our respiratory surfaces are moist. They are moist so they can pick up and void the gases, pick up oxygen, void carbon dioxide, but they have to do it through a moist medium. That is the same for all living things. That respiratory surface represents a tremendous risk, because if it is exposed to the outside, as it would be in a gill or something like that, the loss of water would be tremendous. But in fact, we have adapted a lung which is internal, and as far as possible, we limit the amount of evaporation from the lung. The second adaptation that terrestrial organisms undertake is to modify their waste systems so that they lose as little moisture, as little water, especially, as possible. So there are many different adaptations of anatomy which limit the loss of water in terrestrial environments. And the second thing is that the behavior of terrestrial organisms is often designed to limit water loss. So, for example, in many desert animals are nocturnal, specifically for the reason that it's cooler at night and they lose less water. Second of all, there are all sorts of ways in which their physiology is adapted, so that, for example, they produce very concentrated urine, and they produce very, very dehydrated feces, so that the loss of water is minimal. The third thing is that diet is adapted so that, for example, they eat things which will provide them with water, and very often their digestive systems are adapted to extract as much water as possible. And lastly, there are metabolic adaptations. So, for example, we produce water as a byproduct of cellular respiration itself. When the electrons at the end of the respiratory chain finally reach oxygen, which is their terminal electron acceptor, they form water. And it is possible to conserve that water, to conserve water from cellular respiration, which is what many organisms do. Okay, so on the issue of nitrogenous waste, I've already mentioned, for example, that ammonia is one of the byproducts of protein metabolism. These products are actually extremely toxic, and they have to be avoided. The commonest, end product of metabolism of proteins, amino acids, and things is an amide group, which in itself is very toxic. So in order to get rid of that, the organism is going to have to dissolve it to a certain extent, one extent or another, and in order to avoid it. Now, the waste nitrogen comes actually in three principal forms. First of all, it arises from metabolism, principally of proteins and of nucleic acids, and ultimately forms these amino groups here, NH2. It'll produce compounds of one sort or another with those amino groups attached, which need to be metabolized. These are toxic. And there are three principal products of this metabolism. The first is ammonia, NH3. NH3 I've already mentioned, very, very toxic. Its toxicity is extremely high, but it's very soluble. And if you have abundant water available, then it can be flushed out very, very easily from the body. But the requirement is that you need a lot of water. And so that is not a system which is suitable for any terrestrial organism. And instead, terrestrial organisms have adapted to different mechanisms. The first is, for most animals, the commonest byproduct of the commonest form of nitrogenous waste is actually a substance called urea. There it is here. It's quite a simple compound, but it has two ammonia groups attached to it there. So it's got two amino groups. It has the advantage that it's still toxic, but it's not nearly as toxic as ammonia. It's quite soluble, so it can be avoided in solution, but you don't need as much water because its toxicity is much lower than ammonia. Ammonia, you need to dilute the ammonia to such an extent that it is no longer toxic. Urea with a lower toxicity, you don't need as much water. It's still voided as a solution. And it's, in actual fact, we'll see when we talk about kidney function, this is the major responsibility of the kidney, is to excrete urea. And it does so using various mechanisms, but amongst them, active transport systems, which expend an energy cost. The energy cost associated with ammonia excretion is very, very low. But urea, there is a cost due to active transport of various substances. The third substance is a really interesting one, and that is uric acid. And uric acid is the waste product of reptiles, insects, and birds. Now, the link between birds and reptiles, of course, is an evolutionary one. But in both cases, the reptiles and the birds, they have an advantage in excreting uric acid because uric acid is extremely insoluble. And because it's so insoluble, it has a very low toxicity. You can actually accumulate quite a lot of it. And you can, both reptiles and birds and insects, excrete their uric acid as their principal product. In case of reptiles and birds, you can actually see it. And a bird dropping, the white portion of a bird dropping, that is uric acid. It's a paste of uric acid. All they need is a very small amount to mobilize the uric acid, to be able to literally squirt it out. And they conserve a lot of water by doing that. It doesn't matter that there's some uric acid hanging around because it has a very low toxicity because it's so insoluble. I would just mention something, and that is that in human beings, especially in one or two other animals, but in human beings especially, there is a disorder of the metabolism of nucleic acids. One of the group of nucleic acids are the purines, of nitrogenous bases rather, are the purines. And when there's imbalance of purine metabolism, human beings can excrete uric acid instead of urea. And that accumulates in the joints and it causes a very painful condition called gout. So that is the origin of gout. It's uric acid excretion rather than urea excretion. Okay, so here's what the organism has to do. The organism has to get rid of its nitrogenous waste, and it has to get rid of it in using water. It has to, in getting rid of its nitrogenous waste, it has to do two things. It has to, first of all, make sure that it doesn't use too much water, that it conserves as much water as possible. Okay. And second of all, when it gets rid of its waste, the organism needs to make sure that it doesn't at the same time get rid of stuff that it actually needs. This excretory system has to be designed so that it is selective. It gets rid of what the body wants to get rid of, and it keeps what the body needs to keep. All of these excretory systems, and they go all the way back to even very simple organisms like the flatworms, or the planarias, those organisms have a very primitive excretory system. But in its structure, we see the roots of even the most elaborate, because they're all based on the following principles. First of all, there has to be some way of filtering the body fluid, of extracting a raw filtrate from the body fluids, from the blood, for example. Then that fluid has to be treated so that the useful compounds are reabsorbed and redistributed to the body. The third thing that has to happen is that the system has to secrete those compounds which are toxic or waste products or whatever. And the fourth thing is those products have then got to be voided in some way. So those are the four stages: filtration, reabsorption, secretion, and excretion. There's tremendous amount of variability on how this is done throughout the animal kingdom, but they all basically work on the same kind of principle. And this is exactly the principle that we see in our own excretory system in the kidney. What it consists of is the following. There is some sort of a filtration system. The blood passes through a system whereby fluids from the blood are passed across the walls of the blood vessels and accumulate in some sort of collection vessel. That is the filtration. What is being filtered out are blood corpuscles, which are too big to pass across the walls of these very fine blood vessels. Large proteins like the blood proteins, the antibodies, all of those sort of things, they can't get across the blood vessel walls either. But what does pass across here with the fluid is serum, is a serum which consists of water and many different things dissolved in it, all derived from the serum that the blood was suspended in. So that then passes into a system. The filtrate passes into a system where the useful products need to be reabsorbed. The useful products are things like ions, such as bicarbonate and protons and things like that. These are all useful to the body. And in fact, it is here that these may be kept in balance. So for example, this reabsorption area is often an area where a pH homeostasis of the body fluids is maintained, because you can adjust the body pH by selectively absorbing either protons or bicarbonate ions, things like that. Then the next portion of the tubule is a secretion. That means that substances which the body wants to avoid are allowed to collect inside the tubule, and they're put into solution, and that ultimately forms urine. And that the composition of that urine will vary. It will vary depending upon the organism, upon its environment, and upon its diet and everything else. For example, in desert animals, they will produce an extremely concentrated urine, and they don't produce it very often. So they reabsorb as much water as is physically possible. Organisms like us, which have ready behavioral adaptations to acquiring water, we actually drink a lot of water compared to a desert animal. We do not have to reabsorb nearly as much. And our urine is, we still produce a concentrated urine. We still conserve water, but it is not nearly as concentrated as something like a desert animal. Okay, so here's how it's organized. That simple system. We'll see exactly how it's organized here in our own excretory system. Our excretory system consists of two kidneys. They're here. They lie at the door, closer to the dorsal surface of your body, and just around about the borders of your rib cage. So they'll actually be partially hidden behind the back ribs. They are richly supplied with blood, and they work extremely fast. They filter our blood several times a day. Our whole blood system passes through the kidney several times a day, and it arrives through the aorta and the renal arteries behind here, and the blood percolates through the kidneys and then passes back into the inferior vena cava through the renal veins there. The blood vessels form very complex structures inside the kidney. They subdivide many, many, many times to form very, very fine, tiny knots of blood vessels. So here, this gives you an idea. They look here, and you'll see here, these are arterioles here. And here's a little knot. It's of arterioles inside here, which is called the glomerulus. And that is a knot of very fine arterioles, which pass back and then out into the venous system. Remember that the blood coming in through the arterioles is under pressure from the blood, from the heart beating and the body's blood pressure. The blood pressure here is actually high. These are very, very fine arterioles. So there is actually an increase in pressure inside the glomerulus. Because of that blood pressure, the fluids tend to leak out of the glomerulus, and that is the active filtration. The active filtration is the passage of fluid across the walls of the arterioles. And they are gathered here into, this is called the Bowman's capsule. And it's a collecting vessel. It has tubules which run from it, and you can follow them here. This is called the proximal tubule there. This is called the loop of Henle, and this is the descending limb of the loop of Henle. This is the ascending loop of the loop of Henle. And this area here is called the distal tubule. And the distal tubule feeds into a collecting duct. The collecting ducts all gather together into this system here, which is called the pelvis of the kidney. And the pelvis empties into the ureter, a large tube, a fine tube, which runs to the bladder. And the bladder empties with a large tube called the urethra. But the ureter is what feeds from the kidney's pelvis with all of the urine to the bladder. So they are organized in this way here. If we cut the kidney, you'll see that all the glomeruli are in this area here, and that's called the renal cortex. All of those loops, the loops of Henle, are here in this area, which is called the renal medulla. And there are certain of these. A single unit is called a nephron, and there are certain nephrons with very deep loops of Henle that extend far into the renal medulla, which I'll refer to in a minute. So the other thing to notice is that these tubules, the proximal, the descending and ascending limb, and the distal tubule, are densely surrounded by blood vessels. And that is so that substances which are useful can be rapidly absorbed and returned to the blood circulatory system. So the whole thing is very richly supplied with blood vessels. It surrounds the nephron entirely. So I'm only going to very briefly describe what happens during the process of fluids passing through. This is actually a really complex topic, and we're not going to do it in an enormous amount of detail. But I will attach to the PowerPoints movies for you to watch, and those are required. You must watch those movies, and it will give you a better idea. In addition, in the assignment set for this, there are sections with animations and movies for you to watch and answer questions on. So this is an important section. Okay, you don't need to know all the fine detail that is included in your text, but you do need to know the broad concepts of what is happening. Okay, so here's the glomerulus. We've got the filtrate forming here and being gathered up by the Bowman's capsule there, and now it's passing into this first portion of the tubule of the nephron structure, which is the proximal tubule. Lots of things happen in the proximal tubule. The first thing is that many of the useful ions are actually absorbed right here at the very start. Bicarbonate ions, some sodium chloride, if it's needed, protons, or can be secreted into the proximal tubule. All sorts of nutrients are absorbed if they're present, amino acids, all sorts of things. If they are present in the fluid, they will be absorbed. Useful ions like potassium. So a lot of things happen here, but one thing you have to notice in particular is this is the first absorption of water. And this is one of the major tasks of this nephron structure, is to selectively absorb water and return it to the blood circulatory system. This is the site of a tremendous amount of homeostatic control. And the body responds to changing conditions in blood pH, for example, by changing its uptake of bicarbonate or its secretion of protons, for example, through the proximal tubule. By the time it's left the proximal tubule, what we're looking at in actual fact is a solution which now has lost most of the useful ions and instead consists largely of salt dissolved into water, together with the principal waste product, which is urea. Nowhere along the way here is urea secreted. Nowhere along the way here is it reabsorbed. It is flowing in solution from the Bowman's capsule. So this fluid now enters this descending loop of Henle here. And as it passes down, I just want to point something out to you. And that is that this area here and this area here have a tremendous amount of sodium chloride dissolved in them, secreted through the tubules. You can see it being secreted there and there. And in this area here, it actually, surprisingly enough, has a lot of urea. Why? Well, the reason is that as this fluid, the filtrate, passes through here, it passes through an increasing environment of of increasing osmolarity. So this becomes more, and the environment becomes more and more hyperosmotic, and water passes out very rapidly. So by the end here, it is quite concentrated. As it moves up here, however, up this portion here does not allow water to pass out. But what it does do is it pumps out, it allows salt to flow out first of all, but it actively pumps salt out of the urine. So by the time it reaches back up here, we've already reduced the volume tremendously. We haven't changed the osmolarity that much. It's not that much different to what it was here by the time it gets to here. But we've reduced the volume tremendously and absorbed a huge amount of water. It now passes into the distal tubule here. And in the distal tubule, again, there is selective absorption or secretion. In the distal tubule here, the various ions can be passed into the urine if need be, protons, whatever. More water is absorbed. Salts are absorbed. If need be, bicarbonate can be absorbed, etc. Again, this is an area of homeostatic balance is strongly influenced, especially by hormonal control. The fluid now passes into the collecting duct. As it goes through the collecting duct, more salt is absorbed, and urea is secreted here, again to maintain a high osmotic environment so that water passes out passively. By the time it arrives here, what we end up with is a very concentrated urine, cleared of all the useful substances, containing urea dissolved in it, and that passes now to greater and greater collecting ducts, to the pelvis, and then to the ureter. There's just one other thing I would tell you. In the proximal tubule, and to a certain extent in the distal tubule, but really importantly in the proximal tubule, there are processes which will actively secrete toxins into this fluid. So this here, the proximal tubule, to a certain extent the distal tubule, are areas of purification, where the body actually dumps toxins into the urine. This is one of the reasons why drugs, for example, can easily be picked up in urine. It's because they are actively secreted here into the fluid passing through. Okay, so that is a very complicated story, I know. What I want you to remember about it is its principal task is to conserve water as it passes through the system. Its second task is to void waste materials. The waste materials emerge here with this filtrate. They don't have, the urea doesn't have to be actively secreted into the urine. Drugs they're actively secreted into the urine in the proximal tubule. In this area here, the proximal tubule and the distal tubule, useful substances can be absorbed back into the body. And if need be, things can be secreted into the urine in order to establish homeostatic balance, or in human beings to a small extent to get rid of things like ammonia, that sort of thing. So these here especially are areas of strong homeostatic control. And this is especially true for regulating things like blood pH and even things like blood pressure, that sort of thing, because it's here in these areas that we end up controlling the amount of salt, for example, that is present in the blood. And the salt, the amount of salt we have in the blood also influences the blood volume. The greater the blood volume, the higher the blood pressure. The whole system is designed to absorb as much water as possible and still void the waste efficiently, which it indeed it does. Okay, so I'll just mention that in this area, the loop of Henle, this is where a tremendous amount of adaptation can be seen throughout the animal kingdom. And I just mentioned that desert animals, for example, have extremely long loops of Henle so that they absorb as much water from the urine as possible, and they secrete a very, very, very concentrated urine at the end. Our urine, by the time it emerges here, is relatively concentrated. We have absorbed a tremendous amount of water from the fluid that originated here, but it is not nearly as concentrated as something like a desert animal. And for the rest, it's an extremely efficient filtering system that voids toxins, waste, and yet still manages to maintain and keep all of the useful ions and everything. Okay, so I will put up some movies which will explain it even better. I've given you the background and the skeleton to study it. Please remember, watch the movies, and then also please complete the assignments, because that will complete it all. Okay, and then I will see you on Monday.